Hi guys, my name is Nick. I am one of your SIs for your Biology 5A class, taught by Dr. Joseph and Dr. Yang. Starting this week, we kind of shifted to Dr. Yang being the primary professor, and we are looking at a lot of more of kind of DNA, genetics, and the different experiments that have given us the understanding of these things that we have today. It is kind of a shift from material before, but nonetheless, we should be able to get through everything that we need to. So looking at the learning goals that we have set out for us, we can go through them and explain some of the stuff through the slides and hopefully give you guys a better understanding of a lot of the experiments that were done to push us to the knowledge that we have today regarding viruses and other things. So if we're looking at the properties of genetic material, there are three main things that we have to remember. One is that they carry inheritable information right here. They're capable of being copied, which we see a lot of the time in DNA replication, and they're capable of undergoing change, which is exhibited through things like mutations. And so going forward, we can look at DNA as genetic material, right? But the things that we have to figure out is how did scientists understand that DNA was the primary genetic material as opposed to RNA or protein? So let us look at one of the more famous experiments. Right here is we have the Griffith Experiment in 1928. So this was explained, but an easy way to look at this is that we have two different types of, I guess, pathogenic cell here. We have our S cells and our R cells. Our S cells here, S for smooth, as in smooth because they have the outer layer around here. You can see this orange layer around here. R for rough because there's no outer layer and we were just looking at the actual particles over here. So what we did first is Griffith injected both the S cells and the R cells into different mice to see what the results would be as controls. So the living S cells over here with the orange coat caused the mouse to die as they are the pathogenic agent. While the living R cells that did not have the coat, when they were injected, the mouse remained healthy. And then to see if, I guess, the coat was susceptible to heat or not, he treated the S cells with heat and essentially called them the heat killed ones. And when he injected them, the mouse remained healthy, meaning that whatever it was that was causing this, whatever it was that was causing the mouse to die, was able to be treated by heat, right? And then when we look at this last part here, we have a mixture of heat-killed S cells, which we have seen over here in this control to leave the mouse healthy, and living R cells, which have also been seen to keep the mouse healthy. But when we mix them together, we observed what is now called transformation, and essentially the mouse died as a result of this mixture. And by the end, when he extracted the agent to see what it was relative to what we put in, since we put in heat killed S and living R, turned out that living S were there. And that was the that was the basic idea of the transformation that he was looking at, right? So even though he knew that something was going on here in terms of why the heat killed S cells and the rough cells come together to kill the mouse, he didn't know exactly what it was that underlying factor of transformation. But that was where this experiment happened in 1944 by Avery, MacLeod, and McCarthy, right? And so they took the smooth cells that Griffith had used, the live smooth cells, they killed with heat, and then they extracted the active material. They didn't know what the active material was, but what they did was they used different enzymes to look at it. So RNase essentially degrades RNA. It's a nuclease that works on RNA. Same thing with DNase for DNA, protease for protein, right? So essentially these were the three biggest guesses as to what the active material could have been, RNA, DNA, or protein. And what they saw was the RNA or the cells when treated with RNase, nothing happened. They were still the same, right? The treated cells with RNase, when rough cells were added, no transformation. The treated cells with DNase, when rough cells were added, had a transformation, or no transformation rather. And the protease, when treated with the rough cells, had a transformation. Sorry, let me rephrase that. The no transformation only happened in the ones with DNase. And because of that, they were able to assume that the DNase killed off whatever the active material would have been to cause that transformation, right? Because no transformation occurred here when we added something that's supposed to cleave DNA. So when they saw that no transformation occurred here, they knew that DNA had to be the underlying transforming agent because it was the only thing that responded where the result was different from what Griffith had observed up here in terms of this transformation. So those are those two experiments. And then when we keep going, we can look at the Hershey-Chase experiment, right? So the Hershey-Chase experiment essentially had two parts of it. They were using a bacteriophage, right? And they were trying to see if other macromolecules were the genetic material within this, right? So up top, we have the radioactive sulfur in the proteins of these bacteriophages, right? Bacteriophage has the outer protein coat, and then it has DNA on the inside. So by labeling the coat with sulfur and the DNA with phosphorus, they had two different colors essentially that they could look at. Okay, so once they did that, they allowed the bacteriophages to infect cells, right? And then once they had those cells, they did, that's essentially what this mixing is. We see the bacteriophages injecting what we now know is the DNA into there, and we have the protein coat flying on the outside. But they didn't know this yet because they weren't able to observe it. So once that was done, they did what's called centrifuging. And then we observe two phases here. We have this liquid layer up top called the supernatant, and then the pellet, which has all of this. Right? The supernatant is essentially all the liquid and stuff, and the pellet is the cells that have clumped together at the bottom as a result of centrifugation. So what they did then is that for each thing over here, they took the liquid of this and the pellet of this to see what had changed. And in this one, the radioactivity part was found in the supernatant phase, the liquid phase, meaning at the end of the infection, quote unquote, of the bacteriophage into the host cell, the protein coat was left out. The protein coat was left completely separate from the cell, which is why we observe it in a different phase than the cell. And as you can guess, in the DNA labeled ones, the ones labeled with 32 phosphorus, we saw the DNA actually, the marker actually show up in the pellet phase, meaning that the DNA was what was found in the pellet, meaning the DNA is what was essentially interacting with the cell itself. So by the end of the whole experiment, we saw the protein not interacting, but we saw the DNA injected into the cells over here, and that's why they were in the pellet phase. So Hershey and Chase essentially showed that in this bacteriophage model, the DNA was the genetic material that was being transferred into the cells. All right, so now let's look at Chargaffs rules, right? From 1947 is when we started to figure this out, right? We have percentages. We have 4 different nucleotides when it comes to DNA, right? We have AC, GT, right? And so we know that A and T go together, C and G go together. So it's an easy way, that's the easy way to like sort them into two things first. We know A and T go together, C and G go together, right? The essential idea of this is that whatever the percent of A is has to equal the percent of T in Chargaffs rules. And if you think about it, that makes sense. It just means for every A, there should be a corresponding T. And same thing applying to the GC content, right? For every G, there should be a C that's going with it. If our DNA is completely linked up, if it's going all the way perfect, each A should have a partner, each G should have a partner. Same for T to A and C to G, right? Which is why our equal sign is just going back and forth, right? So if we think about this, if we try to use an example, let's say we know that the GC content of a cell or of our DNA is 60%, right? That would mean that 30% of the content of our DNA is going to be G, 30% of it is going to be C, right? And if we had to figure out our A and T content, all we have to do is think, okay, there's 100% total, right? Our 100% minus our 60% will give us 40% left of nucleotides that we have to account for. So if we have 40% left, and we know that our percent A equals our percent T, all we have to do is divide our 40% in half, and we can figure out the percent of either A or T. So in this instance, we'd have 20% A, 20% T, 30% G, 30% C. And that would make up 100% according to Chargaffs rules. Cool. So let us keep going to look at the structure of DNA. So now that we know about DNA, about kind of how it came to be known as genetic material, we can look at some famous experiments that were done talking about the structure of DNA. So one of the most underrated women in all of science, Rosalind Franklin, did a lot of X-ray diffractions of DNA and found this structure to be essentially what the DNA looked like through the X-ray diffraction. And later on in 1953, Watson and Crick came by, they interpreted this and came up with this general structure of DNA, which we now know to be the double helix. And if you notice, it's harder to draw DNA simply because though it looks like two ribbons, you have to realize it's more of a 3D structure, as in it's going around this like middle line. Because when you draw it flat, the tendency is to draw it like all down one line. But if you'll notice in this picture, it looks like we have two bumps here, and then two bumps here, and then two bumps here, and then it keeps going down just like that. But the reason for that is because it's a 3D structure and it's wrapping all the way around. If you were to take it and flatten it, like let's say you unraveled it around this middle pole and flattened it, it would look like what DNA tends to look like in drawings and stuff. But this double helix structure is represented like this simply because it's in a 3D format here. And like we said, when it came to the double helix structure, they saw it as helical first, but since the DNA molecule was thought to be made up of two strands, as we can see in this general photo, they ended up naming it the double helix. All right, so from there, let's look at some of the properties of the base pairs of DNA. And so we can look back up here, my apologies, right? We can look at some of the properties of our base pairs of DNA, right? So we know, as we said earlier, A goes to T, G goes to C. And if you're wondering what these double lines versus triple lines mean, right? We can see over here, this is a hydrogen bond, right? We have our polar oxygen, hydrogen bonded to a hydrogen that is attached to a nitrogen over here, right? There are only two nitrogen bonds from A's to T's, while GCs have three hydrogen bonds rather. So an AT bond is actually slightly weaker than a GC bond if you were to try and break it, simply because GC has one extra hydrogen bond. And looking at the general structure of this, we can see that we have our nucleotides here in the middle, right? ATCG. We have our sugar over here, right? And then we have our phosphate backbone over here, right? We have our phosphodiester bonds over here, right? Which is one strand goes from 5' to 3', and the complementary strand. Anti-parallel simply means it goes the same, like if you want to think of it as the same numbers. So if we're talking about 5' to 3' here, it still goes 5' to 3', just in the opposite direction. Right, so we have this one, we see this arrow going down, this arrow going up. Even though this is still going 5' to 3', it's in the direction opposite to this. Right, so if you were to look at it straight across, you can say 5 matches up to 3, 3 matches up to 5. All right, and we should note that human DNA is really, really long, right? Our whole genome is 3 times 10 to the 9th base pairs, which is an insane number of base pairs, right? All right, so let us keep going. So when we talk about how DNA encodes information, right, one of the biggest things that we look at is going to be this structure right here, right? The chromosome. So the reason DNA is packed into chromosomes, as you can probably tell, is because human DNA, like it says over here, is 1 whole meter long, right? A meter long in terms of fitting into each individual cell, is incredibly, incredibly long, right? Which is why DNA in all of these have to be packed into chromosomes. Chromosomes essentially take our double helix structure and compress them as far as possible to fit within our super, super small areas, right, in the cell. So if we look here, there's a general structure to chromosomes, right? It'll be gone more into detail when we look at actual chromosomes, but essentially, if you look here, right, we have these blue rings, right? These are called histone proteins. Histones are made up of 4 proteins that come together as one protein, and the DNA essentially wraps around them. It's held around them really, really tight. It's coiled super tight with small spaces in between each histone so that all the histones can come together into this tightly, tightly wound area called the chromosome. All right. And from there, we can look at this last piece of information. Information is stored in DNA, right? And the way information is stored, if we look back to the central dogma, right, we have DNA to RNA to protein. So in our DNA, right, we have a string of base pairs, right? Our A's, our T's, our C's, our G's, right? But depending on the order of that, when it comes to transcription, the different orders of DNA that we have in different areas of our genome code for different types of RNA, which will in turn code for different proteins. As it says here, different sequences, which refers directly to the DNA. If we look at just the top part here, we have a 5' to 3' GTAA GC, or GGC rather. And on this side, we have an ATTA CGA. All right, this 5' to 3', this is 5' to 3'. They're going in the same direction, but they have different sequences, right? And different molecules could refer all the way down to even protein, right? If we have different sequences, they're going to end up as being different proteins. And different information could refer to RNA, where different sequences will code for pieces of RNA that will contain information on making the next step of proteins, and even information that could be used in other parts of the cell. They'll be completely different if the DNA sequences are different. And this occurs all throughout our genome. So for now, that is what we are looking at in terms of the beginning of this. It would be really good to go through these questions and answer them as best as you can. But yeah, thank you for watching the video. Please subscribe and let us know what you think in the comments if there's anything we could change. We would really appreciate it going forward. And thank you guys.